Hello everyone, in this lecture we will see about the giant chromosomes. In some cells at a certain particular stage contain a large nuclei with a giant chromosome. The giant chromosomes are the polytene chromosomes and the Lambert chromosomes. First of all, we will see about the polytene chromosome. It is also called as salivary gland chromosome because it was first found in the salivary gland of the Caranomus larva. The Italian cytologist E.G. Balbering in 1881 observed a peculiar structure in the salivary gland of the midge Caranomus, and this was a long sausage shaped marked by the cross striation and the swelling. At that time he think it may be chromosome, but this body did not show any characteristic like a general chromosome, and hence unfortunately he did not recognize them as a chromosome. But he stated that there is some abnormal body present in the secretory cell of the salivary gland of the Caranomus larva. He did not recognize them as a chromosome. After that, the research is going on, and in 1933, Theophilus Penter earns his and the H. Bott discovered the same structure in the Drosophila, and they recognized them as a chromosome. It is called as a salivary gland chromosome because it is discovered in the salivary gland, and that's why it is termed as a salivary gland chromosome. The Kolar also suggests it as a polytene chromosome because that chromosome also having the polychromonemata or the polychromatids, and that's why Kolar suggests it as a polytene chromosome. Some cell of larvae of dipterian insects like a flies, mosquito, midges become a large having the high DNA content. Mostly this chromosome is found in the larval stage of the insect like a flies, mosquito and midges, and at that time the chromosomal DNA is divided and the sister chromatids are formed, and that sister chromatids arrange one after the other or side to one another, and that's why it become large and also having the high DNA content. These cells are unable to undergo mitosis and die during the metamorphosis, and these larval cells don't undergo the mitosis. Actually, what is mitosis? Mitosis is nothing but the cell division in which it is an equational division in which the single cell is divided into the two cells which having the same amount of the chromosome and the nuclear material. But here the larval cells don't undergo mitosis, and that's why during metamorphosis, means during conversion of larval to adult, that cells die. Such a polytene chromosome located not only in the salivary gland, but it is also present in the other body part cells, Malpighian tubules, rectum, gut, foot pads, fat bodies, ovarian nerve cells. The endomitosis is the one of the characteristic feature of the polytene chromosome, or the polytene chromosome is achieved in the Caranomus larva due to endomitosis. Then what is endomitosis? Endomitosis is nothing but the division of the DNA, but not a nuclear division. Means the replication of the chromosomal DNA take place, but the nuclear division is not taking place. That is the endomitosis, and due to that the polytene chromosome form. The next, and that's why the polytene chromosome of the Drosophila have a thousands DNA molecules, while the Caranomus having the 1600 DNA molecules. The best time to observe the polytene chromosome is the interphase or the prophase of the mitosis. In the polytene chromosome, the chromomer and the DNA fiber are arranged alternately, and that's why the dark and the interbands are formed. The dark band having the large amount of DNA content, that is 85%, and the interband having the 15%. That chromosome also having the cross banding pattern. It is also one of the characteristic of this chromosome, and with due to that we can do the cytogenetic study, and due to that we can differentiate the different species. The cross banding pattern is a specific in a specific species. For example, the Drosophila have the 5000 bands and the 5000 interbands, which repeat the set of the 1024 identical DNA sequences. The maternal and the paternal homologous chromosome remain associated side by side and that is called as a somatic pairing, which is found in this chromosome. Now we'll see the structure of the chromosome. In this chromosome also there is a chromocenter and the 5 strands, 5 long strand and the 1 small strand. From that 5, the 1 long strand form the X chromosome, while the remaining 4 strand forms the 2nd chromosome and the 3rd chromosome. The small strand form the 4th chromosome, which is like a dot, and the male Drosophila, which is found in the female, and the male Drosophila don't have a somatic pairing, and that's why in male the X chromosome is thin and single, while the Y chromosome is fused with the chromocenter. The next is one gene, one band hypothesis. According to this hypothesis, the fixed pattern of the band and interbands in Drosophila polytene suggests that each band have a single gene. Thus, Painter in 1933 and the Bridges in 1936 conclude that Drosophila might have the 5000 genes. It is also believed that each band have a gene and the interbands are the only inert linker region. However, recent data suggests that the band and the interbands have more than the one active gene. Albert in 1989 believed that the bands and interbands may help to keep the DNA organized, isolate the gene from their neighbors and prevent the crosstalk. It also regulates the transcription for the cytodifferentiation. The next point is the chromosome puff or the Bulbiani rings. The swelling of the band of the polytene chromosome, where DNA unfolds into the open loop as a result of the intense gene transcription, means the chromosome puff is nothing but the loop-like structure appear in the polytene chromosome, and which is occur due to unfolding of the DNA, and the DNA unfolding is occur due to transcription. As you know that the transcription is nothing but the synthesis of the mRNA from the DNA, and for the synthesis of the mRNA the DNA want to unfold, and that's why the loop-like swelling occur in the polytene chromosome, and that is nothing but the chromosome puff or the Bulbiani ring. In 1954, Bierman and the Bard studied the structure of the puff. The structure of the puff distribution is different from tissue to tissue, and it is a cyclic or reversible phenomena. Means whenever the synthesis of the mRNA require, at that time it appear like a loop, and whenever not necessary, at that time the loop is disappear. The puffing process involves the accumulation of the acidic protein, unfolding of the DNA for the transcription, accumulation of the RNA polymerase, which is also required for the transcription, and the synthesis of the mRNA, and after that finally the synthesized mRNA is released into the cytoplasm. Thank you.